Good afternoon. I am Shoumo Ganguly, Assistant Professor, Calcutta Institute of Pharmaceutical Technology and Allied Health Sciences, and I am participating in this FDP as participant having registration number 394. I am here to present a small topic as a part of the assignment activity that we have been provided. Now, today I will be discussing something about computer-aided biopharmaceutical characterization and gastrointestinal absorption simulation. It is a part of curriculum of Masters in Pharmacy syllabus that has been given by Pharmacy Council of India. Well, it is a huge topic, but I would like to take you through this short journey just to give you some basics. What is really biopharmaceutical assessment of drugs and why it is very important in different phases of drug discovery and development. Now, when we develop a drug, it is generally a huge process. Like when we are talking of development of vaccine for COVID-19, everyone is talking of clinical trials and all, and it is taking a lot of time to develop either a drug or a vaccine. So why this time is there, or why does it take a lot of time for the pharmaceutical companies to really launch a product? Now it is divided into two main phases, that is early phases and the later stages. Early phases where the drug is developed, I mean to say appropriate drug-like molecules, because all the molecules might not have the therapeutic efficacy, and if a molecule is not therapeutically viable, then there is no purpose in marketing it or rather taking it to a clinical trial phase. And first of all, the molecule has to have some therapeutic effect, and then only it can be designed into a formulation. Now, formulations are of many types. Likewise, we take tablets, that is a formulation. Capsule is a formulation. Injection is also a formulation, but the route of administration is different. Tablet and capsules can be taken orally, and injectables are directly injected intravenously. Now, a growing concern why it takes a lot of time, or rather a huge amount of money for the pharma companies, because it involves a lot of steps. Likewise, in previous time, each and every drug molecule had to go through a series of animal model trials. Now, as times went by, scientists, or rather preclinical formulators, what they are doing, they are trying to do away with this animal experimentation, because it is not possible to test 1,000 of molecules in animals. So what's the way out? Therein comes the importance of in silico tools, which can identify critical factors like drug physicochemical properties, dosage form factors, and predicting drug absorption. Now, why drug absorption? Because human body is a vast system. You never know where the drug is going to get absorbed. It might get absorbed in stomach, it might get absorbed in liver, or it might get absorbed in intestine. Now, what do you mean by in silico? In silico is a term that is used for any activity that is done via computer simulation. Like silico comes from the word silicon, and in this developmental phase there is a huge amount of data. With the advent of supercomputers and very high processing computers, it's very easy to store data and process data. That is why it's very easy for molecular and experimental biologists to get hold of a huge set of data and manage it. And in silico pharmacokinetic parameters can be modeled using different administration route, all using simulation. Now, drug is generally from which pathway it will be absorbed. That depends on various physicochemical properties, physiological factors, and formulation related factors. Why formulation related factors? Because I work in drug development formulations. So when we try to get rather a very fast onset of any therapeutic activity, we just say that why not give it as an infusion or as an injection? Because the drug directly goes into the bloodstream, and once it goes to the bloodstream, it's easily accessible to all the organs. But now, when we are trying to ingest or rather swallowing any formulation like tablet or capsule, The first phase, it goes into the stomach, where the drug degrades. I don't want to use the word degrades, or rather the formulation gets broken up into many small parts, which is then dissolved in gastric fluid, which then moves on to intestine, and thereby the absorption process starts. Now we know that since IV has a rather very fast onset, because the drug is directly getting injected into the bloodstream. Now various approaches have been used. There are various models, that is compartmental absorption and transit model. Now human body is a huge architectural marvel. It is divided into various compartments, and there is various states like equilibrium state and steady state, and there are different dynamic models. That is why there is the compartmental absorption and transit model, that is the CATT model, which we commonly take. This is the very first model devised. Now with the advent of time, and as time progressed and science improved, there came the advanced CATT model, and this model has been commercially integrated into software packages like Gastroplast, SIM, and many other factors. Now, the advantage of in silico simulation tools is that they require very less investment. Now, that is in respect to conduct of animal studies, which require a huge amount of investment. The softwares too are very costly, but that is less when we compare that against animal studies. Number point is that they have the potential to screen virtual compounds. You don't have a compound physically, you just have the structure. By modeling and other factors, you can screen thousands of compounds and evaluate for their properties. Now, theoretical background. Now, the theoretical background is based on biopharmaceutical classification system, and why the prior knowledge of gastrointestinal physiology is very important in designing the Gastroplast softwares. Now this is a model, a CATT model, that is the GI tract consists of 9 compartments linked in series. Using this figure, you can easily understand what are the various compartments, and the arrows represent how the drug flows. Now then comes, there is another way, that is drug dissolved concentration in GI compartment. That is, there are various forms. That is, when a drug enters into a component, it must leave. Likewise, when a drug enters the body, how it is released. The drug does not stay for the end amount of time. The liver metabolizes the drug, which in turn and is excreted through urine and feces. There is another form, that is a paracellular transport, which is a passive form of transport process, which results in the transport of substance across epithelium. There are various factors that can be taken into consider, that is transfer rate constant, dissolution rate constant, absorption rate constant, and luminal degradation rate constant. And finally, there is PBPK model, that is pharmacokinetic, barrier and physiological model. Now, Gastroplast is an ACAT model which requires a lot of input parameters. That's transit time, pH, volume, length, radii, and there are many other factors that need to be known. And effective permeability value is also very important. Now, this is a general simulation strategy. Now, before how to construct a model, without data, you cannot construct a model. So the first part, that is, you need to collect data. Then there are various parameters, and then the model has to be validated. That means there must be reproducibility of result. Now when you explode the model, then there is parameter sensitivity analysis, deconvolution, IV-IVC, dosage regimen changes, virtual trials, and prediction of food effects. Now model construction, modeling and simulation start from data collection. Similarly, mechanistic absorption models require a number of input parameters, and feasibility of using a single simulation or a virtual trial mode enables incorporation of inter-subject variability, and that is also incorporated in Gastroplast and other factors. This is a generalized state. Now comes the parameter sensitivity analysis. Now what is PSA? Parameter sensitivity analysis identifies crucial and critical parameters that limits to absorption or bioavailability of drug, and researchers can save a great deal of time and effort, as it minimizes the loss of resources in pre-formulation process. Now what is a virtual trial? Now, suppose a trial comprises of a number of subjects. Now, Castroplus has the unique property of assuming that the trial is being conducted in various persons of different races. Likewise, the metabolic rate of Indians is different as compared to the African people, and it is varying from the European patients. So, it is very difficult to get hold of all sorts of people together and conduct a trial. That will be a multinational trial. That is very cost effective, but to embark upon that trial, we need to do a virtual trial, and software just specifically allows that virtual trial to take place. Now, fed state and fast state. We know that fed state, likewise, when we have food in our stomach, the body focuses on metabolizing the food very first. Many drugs have an interaction with food, and there are many drugs that has to be taken in the fasted state. Now, this is the chart where fed and fasted, where the stomach pH varies, so does stomach transit time, stomach volume, pH and all. This is a PBPK strategy tool. Likewise, compound specific data, animal model, human model. This is already a correlation. And what is IVC? Now, when we test in lab, that is in vitro, and what is in vivo? When the data is collected from human subjects. Correlation leads to the proper development of a particular pharmaceutical formulation. And IVC GastroFast is a very sophisticated software which incorporates all of the parameters including IVC and all. And that is why GastroFast and GastroPlus TM is one of the most sought out biopharmaceutical characterization software in the current industry. These are some suitable models. And finally, that is the biowaiver consideration is very important, because as per FDA guidelines, then the solubility parameter of BCS and all, and based on the pH, there are many biowaiver consideration. What is a biowaiver? That is the required criteria for approval of drug varies on various physiological factors. And finally, the conclusion part. The various examples presented in this topic demonstrates GI modulase has become a very powerful tool. Parameter sensitivity analysis is very useful, as it allows identification of critical factors without really carrying on the experimentation. An in vivo, in vitro, in silico approach is successfully used to identify dissolution specifications, and there are many various other factors. And finally, there are shortcomings. Certain gaps are also there, but with time, the things can be modified as well. That's all. Thank you for listening, and it has been a pleasure to be a part of this. Thank you, everyone. Thank you.